Hello everyone. I am Aswathy K, advised by Dr. Karthik Raman of Department of Biotechnology, IIT Madras. Today I'll be presenting our work on constraint-based method to identify function-specific minimal microbiomes from large microbial communities. Microbes are present everywhere on the planet. Microbial species do not exist alone in nature. They form communities and survive by interacting with each other. Depending on the types of species present in the community and also on the environmental factors, the members of the community exhibit competition and cooperation. For instance, certain members of the species may compete with each other for certain metabolites, whereas certain other species may cross-feed and cooperate in the community. Depending on the compositional makeup and also on the types of interactions, the community exhibits biological functionalities. The microbial community present in the human gut, called the gut microbiome, is an essential part of human digestive system. The gut microbiome is capable of producing amino acids, vitamins, and is capable of digesting dietary fiber, which is otherwise indigestible by the human body. The gut microbiome breaks down the dietary fiber to short-chain fatty acids, which are essential for human health. For instance, butyrate, which is a short-chain fatty acid, is known to induce apoptosis. It helps develop intestinal barrier, regulate immune system, and so on. The gut microbiome is a very essential part of human health. Disruption of healthy gut microbiome is known to cause several disease states such as obesity, type 2 diabetes, inflammatory bowel disease, and so on. All the species present in a large community do not contribute to a given functionality. Oftentimes, it's possible to identify a smaller community, which is a subset of the large community, that's capable of a specific functionality that we're interested in. All the species of a large community are not capable of digesting the dietary fibers. Some of them can do that, and they are regarded as keystone species of the community because they are the providers to other species. A lot of the species, although dissimilar in nature, they may have functional redundancies. So because of all these reasons, the minimal microbiome, a smaller community that we are identifying, it need not be unique. It's possible to identify multiple minimal microbiomes for a certain functionality that we are interested in. Knowledge of minimal microbiomes is useful for designing treatment strategies for certain diseases caused by disruption of the gut microbiome. For instance, Clostridium difficile infection is treated using fecal transplant method. Instead of that, we could cultivate a minimal microbiome and administer that to the patient. So this could probably help us prevent any unintentional transfer of pathogenic species during fecal transplant. It's also useful for bioengineering applications, for bioremediation, wastewater treatment, and so on. In such situations when any perturbation of the microbial community occurs, the concept of minimal microbiome can be used to overcome the problem. So we are presenting a systematic mathematical method to identify a minimal microbiome from a large community that's capable of a certain functionality. By functionality, we mean production of certain metabolites, maybe certain short-chain fatty acids. Our method is top-down and is constraint-based. Here is a formulation of our method. In first step, we find the maximum growth rate of the community. This is a joint flux balance analysis, and it's a linear programming problem. In step 2, we find the maximum metabolite production for the growth rate obtained in step 1. This is a flux variability analysis, and it's again a linear programming problem. The growth rates and the metabolite production rate obtained in these steps are our constraints for identifying the minimal microbiome. In the third step, we systematically delete species from the community. This involves multiple linear programming problems because once an organism is deleted, we check for satisfaction of growth rate and metabolite production constraints. So that involves solving linear programming problems. When the constraints are satisfied, if the constraints are satisfied, that organism is removed. If not, the organism is retained and we move on. In the step 4, minimization of L1 norm of membership vector of resultant smaller community is done. This is a mixed integer linear programming problem. The size of the MILP is the actual size of the large community minus the number of deletions done in the third step. Step 3 helps reduce the size of the MILP problem. Apart from that, it's also needed to identify multiple distinct minimal microbiomes because the composition of the minimal microbiome is determined by the sequence in which deletions were done in the third step. This is the formulation of joint FPA. It's a well-known formulation. The objective is to maximize the sum of biomass fluxes of all the species present, subject to constraints on stoichiometry and bounds on the flux values. The overall exchange flux from the community is the sum of the exchange fluxes of all the member species. This is a coupling constraint. It ensures that there is no metabolite exchange in the absence of growth. So in the absence of this constraint, we might get spurious results in which species exchange metabolites without growing. After this FPA, we conduct flux variability analysis. For the growth rate obtained from here, we find out what is the maximum possible metabolite production flux. And those form the constraint for our MILP problem. And this is a formulation of MILP. The objective is to minimize the L1 norm of the membership vector. The components of the membership vector are 0 or 1. It indicates the presence of the member in the community. If it is 0, it means that the species is not present in the community, and if it's 1, it means the species is present in the community. Subject to constraints on stoichiometry, flux values, upper bounds and lower bounds. Then coupling constraint, the flux values are the continuous variables and the community membership vector components are 0 and 1, which are binary integers. These are the constraints imposed by the large community from the previous steps. The exchange flux of the SCFA in the minimal microbiome has to be greater than or equal to a fraction of the exchange flux of the SCFA of the large community. The biomass flux in the minimal microbiome has to be greater than or equal to a fraction of biomass flux of in the large community. We have this coded and it's available on our GitHub page. The code is quite flexible and allows the user to input a lot of the parameters that were discussed earlier. The mandatory inputs for running the algorithm are a community model and the constraints on exchanges to identify the diet of the community. The optional inputs are growth rate fraction, SCFA fraction, the coupling constraints. Here is the constraint for functionality. What kind of functionality are we looking at? Do we want to maximize a certain metabolite or do we want to maximize some of a few metabolites? That can be provided as the constraint. Then maximum number of the organisms for the MILP. This will determine the number of deletions to be made in step 3. Total number of iterations the code should run. Running it once will yield one minimal microbiome. So to identify distinct minimal microbiomes, we have to run it multiple times. The number of rounds, the deletion has to be taken. Then the original growth rates while finding maximum SCFA production. So this is the fraction for considering while doing flux variability analysis. So we know that suboptimal growth rates are favorable for SCFA production and that they are also realistic. So we can tune that parameter to obtain the realistic values for SCFA production. Sequence for deletion in step 3, it's otherwise randomly generated. Here is the result of analyzing a 9-member community for identifying the minimal microbiomes. This is the 9-member community. Among these, Bacteroids Cytoitomicron and Faecalibacterium prosnitzi are the only two organisms that are capable of digesting dietary fibers. So these are the keystone species in this community. On the functionality of butyrate production and parameters as given here, on high fiber diet, we observe that the minimal microbiome consists of FP and E. coli. When the optimum growth rate fraction is changed from 0.9 to 0.90, this now increases the production of ACFA by the large community. So that imposes a more demanding constraint on the minimal microbiome. It's now expected to produce more ACFA. So it has more number of species now. It includes both BT and FP in this case. When we increase the growth rate fraction from 0.8 to 0.9, this is again a demanding constraint because the minimal microbiome is now expected to grow at a higher rate and also produce SEFA. We need 3 organisms, and for this case, we were able to identify 2 distinct minimal microbiomes. And it may be noticed that each of these microbiomes consists of either BT or FP, which are the keystone species in the community. On the same parameters, when the diet is changed to Western diet instead of high fiber diet, it's observed that FP and EC form the minimal microbiome. Here is a result of analyzing a 25-member original community on high fiber diet for butyrate production on parameters as described here. We were able to identify 18 minimal microbiomes, and here is a plot of the frequency of occurrences of all the species present in the large community in the minimal microbiomes. We know that bacteroid species are all capable of breaking down dietary fiber. They're all the keystone species in the community. That explains why the occurrence of bacteroid species are high. Some of the species are functionally redundant, and that's why some of these are occurring at a lower frequency because they can be replaced in the minimal microbiome. They can be interchanged in the minimal microbiomes. To summarize, we have developed a constraint-based method to identify minimal microbiomes which are non-unique of a large microbial community. The mandatory presence of species capable of digesting dietary fibers is observed in the minimal microbiomes. The identified minimal microbiomes can be mutually exclusive. The algorithm can quantitatively predict metabolic fluxes. This approach is reasonable for a coexisting community because of the constraint-based approach used. The gut microbiome is known to be a coexisting community, so it's reasonable to use it for the gut microbiome. Regulatory effects and spatial organization are not considered in this algorithm. If these are to be included, then additional constraints will have to be added. To conclude, large communities can be reduced to function-specific minimal microbiomes. Constraint-based approach to identify them is being presented. The algorithm is designed to be flexible to accept several parameters as user inputs. The minimal microbiome code is available on our GitHub page. I would like to acknowledge Professor Karthik Raman for guiding me, IBSE for the postdoctoral fellowship and the travel grant, and all the members of Raman Lab for the suggestions. Thank you.